So we are presenting on the endocrine systems of the termite. There isn't a whole lot of information available on the reproductive hormones in the termite, but what we were able to find is that there are juvenile hormones, pheromones, and activosteroids. So juvenile hormones are steroids. They are extremely important for the development of the yolk in the female termite. They also inhibit the reproduction of termites later in their development when they're larvae and other. Pheromones are important for attracting males during sexual reproduction. All of these different hormones are neuroendocrine hormones. They are released by the cerebral ganglion in the termite, and then they travel through the hemolymph, and they leave the termite at different spots on the body. Within colonies, only the king and the queen are sexually mature. And so the upper castes, like the soldiers and the king and the queen, are responsible for releasing juvenile hormone to prevent the lower castes from becoming sexually mature. So pheromones are secreted by the sternal gland, which is at the back of a termite's abdomen. The female releases this when she's preparing to mate. This will attract males. The male then touches the female's abdomen near the area that the pheromones are released from. And in that way, the female will begin to release eggs that can be then fertilized by the male. Juvenile hormone is secreted by the female termite's corpus allatum, which is in the cerebral ganglion, and it is secreted in response to the male termite stimulation of the membranes in the abdomen. This causes the release of fatty acids, which allow for the development of the yolk of the female's eggs. Juvenile hormone is also secreted by worker and soldier termites. The mechanism and stimuli are not entirely clear, but they do prevent the maturation of larvae and of the lower termite castes.
Termites can reproduce any time when the environment is favorable. The climate plays a big role during this process. They usually mate during rainy season, where it's hot and humid. When there is adequate resources available for them to reproduce, some workers, called alates, molt into winged adults and intermingle with termites from different colonies. We call this process swarming. This process increases genetic diversity. When male and female alate form a pair, they land and break off their wings. Then the pair prepares a nest in a small hole or depression near soil or wood. They seal their nests using their own saliva, gathered soil, and their own wastes. Then mating occurs, then the female lays eggs. After the establishment of a colony, mature queens can lay eggs up to 1,000 times per year. Their fertility also depends on the resources available.
So juvenile hormone in termites activates the metabolic pathway known as MEKRE93. Once it's released, juvenile hormone is transported through the hemolymph with the aid of high-affinity juvenile hormone binding proteins. These proteins help to protect the juvenile hormone from being degraded by enzymes. Juvenile hormone binds first to an intranuclear receptor called methylpurine tolerant. Binding this receptor initiates dimerization of MET with another protein known as thymine. The subsequent JH-MET-Ti complex initiates transcription of the Krupel homolog 1 gene that represses the E93 gene, otherwise known as the adult specifier gene, and thus inhibits metamorphosis. The complexed juvenile hormone is then able to be degraded by special enzymes such as JH esterases and JH epoxide hydrolases, and subsequent reduction of juvenile hormone titers then promotes metamorphosis. So for a short summary, juvenile hormone works to repress adult morphogenesis by activation of the MEKRE93 metabolic pathway to ultimately inhibit the E93 gene.
Asexual reproduction is used by termite queens in order to produce replacement queens. Termites also use outbreeding, where males and females from different colonies mate together, therefore increasing genetic diversity. Inbreeding also often occurs between related termite kings and queens, or primary reproductives within the colony. The rest of the information found was about cockroaches, which is a similar organism to the termite, since there was very limited information on fertilization in termites. In terms of sperm and egg production, the sperm are produced via spermatogenesis, while eggs are produced during oogenesis. During copulation, the sperm are passed to the ejaculation duct and mixed with a nourishing fluid to become a spermatophore, which is passed to the vestibulum of the genital pouch in the female. Here, the sperm can be released from the spermatophore to fertilize the eggs in the ootheca. which is a mass of about 16 eggs. Once the eggs of the ootheca are fertilized, the ootheca protrudes out of the female's abdomen until it is deposited. Embryonic development occurs in the ootheca as the eggs undergo blastula and gastrula formation before being developed into an embryo. Gestation, the time between conception and birth, lasts about 26 to 30 days for termites.
When a termite hatches, the nymph is pale and blind. The nymph can move, but overall they are still quite vulnerable. They are entirely dependent upon other termites in the colony to survive. Once enough nymphs turn into workers, they will then take care of the newly hatched nymphs. The picture shows the life cycle of a termite. At the start of a new colony, the king and queen, but mainly the king, take care of the young. As the colony grows, the once baby termites grow into worker termites who will then take care of their siblings. The king and queen can spend more time and resources to produce more babies. The picture on the right shows the size difference between the king and the queen.